All right, it is February 25th, 2021. Moving on to a new set of notes called Pre- and Postnatal Testing and Tumor Markers. So, first let's talk about amniotic fluid. This fluid contained in the amniotic sac, which envelops around a fetus. So that's what amniotic fluid is. It's there's a sac that develops around a fetus, and this is the fluid that wraps around the fetus. So, collected by amniocentesis. Amniocentesis. That's that's what it's collected by, and that has three general purposes. One, as a guide to possible action needed in erythroblastosis, fetalis, isoimmune hemolytic disease. That's the first thing. Second thing is a guide to fetal lung maturity. So they can test that amniotic fluid to see how good the lungs are. Number three is a guide to genetic counseling. That's cool. I think that's cool. Genetic counseling. Anyway, and let's talk about analysis of an amniotic fluid. Alpha-Fetoprotein. I remember hearing about this several times. It is indicative of neural tube defects, spinal bifida, I'm going to try to pronounce this word, anencephaly. Okay, so alpha-fetoprotein, neural tube defects, spinal bifida, encephaly, brain defect, hydrocephalus, excessive fluid affecting brain, and others. So it seems like this amniotic fluid with alpha-fetoprotein, a lot to do with the nervous system, because spinal bifida, the spinal cord, that's nervous system, and then you can have encephaly, which is again brain defect. So, and hydrocephalus, so excessive fluid in the brain. So it's all to do with this nervous system brain stuff. These allow embryonic protein to be washed from the open tube, ending up in the amniotic fluid. Alpha-fetoprotein peaks in about the 13th week. So, 13th week of pregnancy, alpha-fetoprotein will peak. And let's see here. If needed, amniotic samples can be taken, but usually not until an abnormal result is obtained from the less invasive serum sample, which is a good screening test since AFP does cross the placenta. So I imagine they kind of want to not do, this amniotic fluid, getting that sample probably would try to avoid that. And it's a good thing that the AFP crosses the placenta, this alpha-fetoprotein. So this is generally done between the 15 and 20 week range, with 16 to 18 being optimal. So that's best to do it between 16 and 18, but can, generally 15 and 20. Alpha-fetoprotein is expressed as a concentration and multiples of the median, MOM. I remember on the worksheet, she asked us a question, what is MOM? And the answer was multiples of the median. So that's, and multiples of the median, MOM, that's where alpha-fetoprotein is expressed as a concentration. So the normal MOM, multiples of the median, is 0.5 to 2.5 MOM. So Low levels are seen in Down syndrome. So if you have a low MOM, multiples of the median, seen in Down syndrome. High, seen in neural tube defects. Alpha-Fetoprotein elevations are seen with hepatic and testicular cancers as well. That's so liver, hepatic, testicular, testes. Cancers with those two, the AFP, alpha-fetoprotein, will rise. So, Down syndrome is the most common fetal chromosomal abnormality, an extra chromosome number 21, trisomy 21. So, and I think probably everybody's, probably heard of Down syndrome in their life. Maybe even know someone that does have it. But it's the most common fetal chromosomal abnormality, and has to do with an extra chromosome, just an extra chromosome. Number 21, trisomy 21. So, it is characterized by several mental retardation and other associated physical deformities. Yes, it is. Yes, it is. Best test is chromosomal analysis called karyotyping, which, so if you want to test for Down syndrome, you're going to do something called karyotyping. That's what you're going to do, is karyotyping. Let me underline that. Karyotyping. That's what the test that you're going to do. So, karyotyping. Best chromosomal analysis. So, which requires fetal cells generally acquired through amniocentesis. So, karyotyping, which requires fetal cells generally acquired through amniocentesis. So, which has a 1 to 100 risk of complications. Whoa. The best non-invasive test are decreased AFP. In parentheses here you have below 0.5 MOM, and increased HCG levels and decreased levels of unconjugated estriol measured in the maternal serum. So, and there's a lot of black and bold letters here, so imagine this sentence has a good chance of being on the test. So let's underline it and say it again. The best non-invasive tests are decreased AFP, MOM below 0.5, increased HCG levels, so that's human chronic gonadotropin, and decreased levels of unconjugated estriol measured in the maternal serum, all utilized in conjugation with each other, so they're all together. All three tests provide a better predictor than any one alone. So it looks like, don't just do one, do all three, do them all. Those determined to be high risk may have the more risky amniocentesis performed, and we can read the book. So, assessment of isoimmune hemolytic disease. Isoimmune hemolytic disease. Erythroblastosis fetalis. It looks like we're going to be talking about red blood cells, just by the name of this. Hemolysis maybe, isoimmune. So we're going to be assessing this. Assessment of isoimmune hemolytic disease, erythroblastosis fetalis, in short called HDN. Alright, so this results from blood type incompatibility between mother and fetus. I was right. So it looks like the mother and the fetus will have a different blood type, and that's more common than what you think. Maternal blood group antibodies, IgG, cross the placenta and destroy the fetal red blood cells. The worst cases are generally due to Rh-ABO incompatibility in the most common type of HDN and is less severe. So, okay. When antibodies attach to the fetus cells, the fetal reticuloendothelial cells phagocytize the injured red blood cells, causing an increase in bilirubin. Okay, I see what's happening here. So you have the mother's antibodies attaching to the fetus cells. So the reticuloendothelial cells phagocytize those cells, and the cells that they phagocytize are going to like blow up. They're going to die, which releases bilirubin. So you have a bilirubin increase from that. Some bilirubin will end up in the amniotic fluid. So this can be measured to estimate fetal risk danger. So I imagine if they test that amniotic fluid and there's an increased bilirubin, they can tell that the fetus's cells have been phagocytizing those cells with the antibodies, and so a lot of that's been going on and causing that bilirubin to rise. Some bilirubin will end up in the amniotic fluid. Yeah, this can be measured. Oh yeah, I've already read that. So, and it looks like in blood bank, also called immunohematology, we're going to study that in more detail. I'm already like 12 minutes. Daggone. Assessment of fetal maturity. If it becomes necessary to terminate a high-risk pregnancy, IHD, DM, preeclampsia, edema, hypertension, proteinuria, premature labor. Okay, so that's all things in parentheses. Usually by C-section or induction, it must be determined if the fetus has a sufficient degree of lung maturity. If insufficient lung maturity is present, the infant may experience serious respiratory difficulties known as respiratory distress syndrome, or DS. Alright, so I'm going to end it right there as I am getting closer to 15 minutes, and I believe YouTube told me I can't post longer than 15 minutes. That's how I understand it.